Hello, everyone. I'm Brian Waterhouse. I'm happy to welcome you all to the second quarter scientific webinar hosted by the Safety Scientific Working Group of the ASA. Today is the second part of a two-parter. We're focusing on benefit risk assessment processes, visualizations, and more. Okay, there it is. So just a reminder that the ASA BioPharm Safety Scientific Working Group has a public-private partnership with the FDA. We're split into three work streams. The work that we're going to be highlighting today comes from work stream 1, and in particular from the Benefit Risk Assessment Tool Suite task force, which is one of 4 task forces within the work stream. Now, the BRATS, as we like to call ourselves, is really working in conjunction with the Benefit Risk Assessment Planning task force, focusing on benefit risk. And as part of our goal of creating a tool suite to assist in benefit risk assessment, we are talking amongst ourselves with what are the best practices and what do people do within each individual company. So as I mentioned before, this is the second of a two-parter. Previously, we had people from AstraZeneca, Tim Sullivan and June Park, as well as 2 folks from UCB, Mike Colopy and Chen Chen, give a presentation in May. That's available on our YouTube. And today we will be going over the agenda as soon as I get the slide to move. There it is. Okay, great. So we're here talking about the agenda. This is the opening and the introduction. Martin will be giving us a presentation about BRATS, followed by Danae, who is going to be talking about the GSK quantitative benefit risk assessment, a fictitious example in multiple myeloma. This will be followed by a panel discussion with Tim Sullivan, Mike Colopy, and Hong Yang from the FDA. And then we'll have closing remarks. So without further ado, let's get started. And Martin, please take the ball and let's get rolling.
Okay, perfect. Brian, thanks a lot for the nice introduction. So I will share my screen. Can you see it?
Yes, you can see it now.
Perfect. Good. As you've seen on the slide, I'm Martin Gebel. I'm a statistical expert, safety and benefit risk at Bayer. And among other things, I'm also a member of the BRAP and the BRATS initiative as part of Workstream 1. Okay, and today I will present about benefit risk assessment using BRATS, which is an internally at Bayer developed Shiny application for structured benefit risk assessment. So my presentation will be split into 2 parts. The first part, I will shortly show how we do benefit risk assessment at Bayer, mainly the process and when we do what kind of analysis. And in the second part, I will introduce the BRATS app. So let's get started with the benefit risk assessment at Bayer. So our approach to structured benefit risk assessment is as follows. We have the so-called COBRA, which stands for Core Benefit Risk Assessment, as a key process, which is applied in every project. And also in every project, we have a benefit risk management team. I will show you further detail on those in the following. And for each project, we have also a so-called statistical benefit risk advisor in place as the stats responsible. When it comes to submissions, we have the FDA benefit risk framework and the EMA effects table usually as submission standard elements. When it comes to semi-quantitative analyses, they are not necessarily done in all projects, but rather depending on the project requirements when needed. We have some method groups within Bayer. We have a cross-departmental group called Benefit Risk Assessment and Communication. This working group is led by pharmacovigilance. And the idea is that this is a cross-functional team to share knowledge, ideas, and project experience, and also consult the project teams. Additionally, we have within statistics, the so-called Biostatistics Innovation Center, and one subgroup is here dealing with benefit risk. And among several other things, we recorded several benefit risk webinars on statistical methods and frameworks, and these are available to the Bayer community. When it comes to the projects, we have assessed the benefit risk management team, and this is led by the global safety leader from pharmacovigilance, and also holds members from several departments, meaning clinical development, medical affairs, preclinical, regulatory affairs, statistics, commercial and market access. And additionally, in case we have a combination product, also a medical device expert is included in the benefit risk management team. As said, the COBRA, the core benefit risk assessment, is our process for doing those. And this is one of the tasks of the benefit risk management team. It consists of 9 elements. The 5 first elements you see here are supposed to be very lean and concise, describing the general setting of the project. And the main focus is on the following 3, the key benefits, the key risks, and the risk management needs. And finally, on the integrated benefit risk assessment, which is supposed to summarize all 8 modules, which are part of the COBRA. And this is usually started as when we go first into man or shortly before that. It's the idea that across the medical product life cycle, this COBRA document or process is evolving. Of course, the more and more information you assess based on studies and things like that, the further information will be added into the COBRA. And also across the medical product life cycle, the idea is that several documents like the RMP or the submission dossier or the label can profit out of the COBRA and take elements out of there. Okay. When it comes to statistics, we have 2 roles at Bayer currently. One is the statistical expert for safety and benefit risk. This is a dedicated role in statistics, which I am currently holding. And the idea is that I actively contribute to internal and external working groups, like the BRAP and BRATS initiatives and trainings, and also guidances regarding safety and benefit risks. Internally, I consult and try to align the conduct of benefit risk approaches across projects. Additionally, on the project level, we have the so-called statistical benefit risk advisor. The SPRA is assigned on the project or compound indication level around start of phase 2 or at the beginning of strategic safety discussions for a project. The SPRA collaborates with the global safety leader and the global clinical leader, and this is a member of the benefit risk management team I showed you earlier. He or she proactively drives statistical safety and benefit risk considerations and evaluations throughout the medical product lifecycle. He or she initiates or contributes to early discussions on advanced safety analyses of individual trial data and also integrated databases, and should serve as a statistical advisor for all safety and benefit risk topics. For example, the support of risk definitions and the benefit risk assessment strategies throughout the medical product lifecycle. And in case we have an aggregated safety assessment plan, an ASAP, in a project available, he or she should co-author the relevant parts from the statistical side. So summing the benefit risk assessment at Bayer up, it's still a project-specific decision how we apply benefit risk assessment. In general, of course, we have a structured process in place, the COBRA, but the type of analysis depend on the particular project-specific situation. The common approach is still to do qualitative assessment. In case we have a project where there are similar types of outcomes for benefit and risks, then we usually do a semi-quantitative analysis, and these can either be done with SAS, creating technical reports, in case these are part of a submission, but also can be done or additionally be done with the BRATS app, which I will show you in a minute. And upon request, we did for some projects, and likely will do also in the future, do fully quantitative analyses, including, for example, patient preference data and physician preference data. Okay, this is more or less in a nutshell how we do benefit risk assessment at Bayer. And now I will continue with the second part of my presentation, the BRATS app, which we internally developed over the last years. So the BRATS app is an app for structured benefit risk assessment of clinical studies. And there are two main elements, the forest plot and the value tree. And it can be applied throughout the whole medical product life cycle. The one element, the value tree, provides an interactive visualization of benefit and risks, especially for the planning of the benefit risk assessment. And the second element, the forest plot, provides estimate for all relevant efficacy and safety outcomes in one graph. And here we present pre-calculated estimates for the comparison of verum versus control. And it is possible to show hazard ratios, odds ratios, risk differences, relative risks, or also, which is commonly used in semi-quantitative analysis, the so-called excess number of subjects based on either cumulative incidence differences or risk differences for a hypothetical population. Additionally, we can include in the forest plot the event and patient counts, or also number needed to treat and number needed to harm. And the forest plot can be used with single study data or also with pooled data. So now jumping a bit to further detail regarding the value tree, this is an interactive color-coded visualization of the benefits and risks for a drug to support the benefit risk analysis planning and also the later assessment. It's usually useful for brainstorming about the key benefits and risks throughout the medical product life cycle. Ideally, it should be started early in drug development, for example, around beginning of phase 2. And the idea is that you can or should update it at each subsequent decision gate, or in case new data about safety or efficacy are available. When it comes to BRATS, the benefit and risk can be displayed as specific measurable outcomes in the distant branches of the tree. The benefit criteria will be coded by default in green and the risk in red, but you can also adapt this if you want. And in line with the RMP wording, we can also separately show it potential risks in orange. And after you've set it up, you can save the graph in an HTML format. Here you see a screenshot, and several will follow, of the BRATS app, how the setup of the value tree works. You can select between the two types, benefits and risks, or separation of risks into identified and potential risk. You can change the colors if you want. You then can add nodes to the tree or remove them if you want to change something, or shift them if you want to put it somewhere else. And after you've set up your value tree, you can download it as an HTML file. Here's an exemplary output of a value tree created by BRATS, where you see the benefits and risks separated into several categories. And as an alternative, we have this second style where we separate the risks into identified and potential risks as set in line with the wording for the categorization of the RMP. Okay, this was the value tree. Now I will talk a bit about the second element of the BRATS app, the interactive forest plot. The forest plot is based on CSV file, which is supposed to include the estimates and further information about these aggregated estimates, which are supposed to be presented in the forest plot. When you uploaded the data, the efficacy and safety parameters can be interactively added and removed from the forest plot. And ideally, the aligned value tree is the basis for the selection in the presentation of the forest plot. The estimates for the various efficacy and safety outcomes of interest can be compared based on the same data scope and population, and this can be done overall and also by subgroup in case they are of interest, or for example, for a pool by study. And with that, the BRATS app supports structured benefit risk assessment with these semi-quantitative analyses. And now showing you how this will look in the app. Here you see again how you start the app, how it looks like. So first you upload the CSV file. There can be some country-specific differences between the CSV files, which you can here adapt before the upload. So for example, how decimal places are used either with point or comma, and how quotes are used, so you can adapt this to your particular CSV file. And after you've done that, you can then upload one or more files in case the data is separated into several CSV files. The limitation is up to 40 megabytes currently, which is usually sufficient because we are only uploading aggregated estimates here. As key elements of the file is an indicator for the type of estimate you are uploading, the analysis set the data is based on, and of course also the actual effect estimates and the corresponding confidence intervals. Additionally, there are optional elements, meaning the number of events and patients for verum and comparator, studies, stratum, and or subgroup variables, the data scope in case this is stored in a separate variables, and also NNT and NNH. When it comes to the estimates, which can currently be presented in the BRATS app, there are several options available. You can present excess number of subjects with event, for example, based on the cumulative incidence differences or risk differences. But you can also show other estimates like risk differences, hazard ratios, odds ratios, and relative risks. Additionally, in case the data is available in the CSV file, additional elements can be shown. The actual estimates, of course, they should be available as numbers additional to the graphical presentation, but also the event numbers and patient counts and NNT and NNH in case they are available. So how does this work in the app? Here you can see again the interface. You identify the analysis set, the data scope in case it's available, choose the study in case you have maybe more available in your file, and also a stratum in case you want to show results by stratum. Then you select the estimate of interest. It can also be possible that you have several available. Identify the efficacy outcomes of interest and also the safety outcomes of interest. And later on, in case you want to do analysis by subgroups, you can also click here and identify them for the presentation. But first, going into a general presentation, as said, you identify the estimate here, for example, incidence rate differences. You select the efficacy outcomes of interest and then the safety outcomes of interest and push apply. And then you will see here an exemplary forest plot showing first the outcomes for efficacy with, in case you want to see the numbers as estimates, and also the patient and event counts, and similar for safety, the same. In case you also want to see a presentation by subgroup, you can pick one of the subgroup in the subgroup tab. And in case you did this, an additional sub-level selection appears where you then identify the sub-level you are interested in. You can either pick one or also pick more if you want to maybe compare the results by subgroup initially in one plot. So if you, for example, now pick here sex and want to compare in the plot the males and the females, the plot looks like this. So you see for both outcomes, for all outcomes, the results for the two subgroup categories, and this follows then consistently for all the selected outcomes so that you directly can compare those. Okay. In case you then also only want to see one particular sub-level, you can also select this, of course, and this would then look like this. You see just the results for this chosen sub-level category female for the selected outcomes of interest. Okay, what you can additionally do in case you want to optimize the presentation of the forest plot a bit, set these estimates, counts, and NNT and NH are optional. You can either click or unclick them. You can change the automatically created title based on your selection in case you want to add something differently there. You can change the sorting of the outcomes of interest either as input or alphabetical or based on the effect size. You can zoom into the plot in case you're particularly interested in a certain area, or maybe your laptop is a bit small or whatever. And also in case, especially when you have maybe an outcome with huge confidence intervals in a subgroup, for example, you can also limit the x-axis. Okay, this was, sorry, I'm jumping back for a second. This is what we are currently having in our main branch available at GitHub. What we developed over maybe the last year or so was an addition to our app. We tried to implement a calculation of the outcomes or the estimates for the outcomes directly based on ADAM data sets, ADTT and ADSL. And this is, I think, for a week now available on GitHub as well. It is still a beta version. So in case you want to try it out, please use it with caution because we are still testing and there are still some minor bugs in, but we wanted to make it available. As I said, it's in a different branch, the beta branch, but we hopefully after having the testing finalized soon will update our main branch as well. And here's how it looks like. You upload your ADTT data set, and additionally you can also update your ADSL data set in case there are like subgroup variables also in you also would like to consider in your analyses. And then you select your treatment variable, your identifier for verum and for comparator. You select the variable which specifies the outcome, select the outcomes you want to calculate your estimates for, or the outcomes. Add the analysis value variable, which is usually the days variable, or the relative days variable. An indicator for the analysis set and how this is specified, the variable which provides you the subject identifier, and this one is the only optional variable, the data scope variable, because sometimes it's stored separately or sometimes it's combined with the outcome variable, so this is not necessarily required. But in case you have it in your data set, please also use it here. Additionally, you can calculate estimates by subgroup. Then you have to click here and identify the subgroups you want to do your calculations for. And you can also additionally filter your data set in case this is needed. Then you have to specify your event identifier and select which outcome of this variable identifies the patient which actually have an event. And finally, you select the estimate of interest. In case you want to calculate, as it is here, cumulative incidence differences, you also have to additionally identify the time point where you cut and calculate the cumulative incidences. And additionally, for some like hazard ratio or incidence rate difference and risk differences, you can also apply a stratification by several variables of interest. And after you've selected all the settings here and variables, you push on calculate, and then after you've got your results, you can either send the results directly to the app and take a look at the forest plot, or you can also save your data as a CSV file so that you have it available at a later point in time. Okay, here's what I've more or less just told you, already some repeated. So what we currently have available are hazard ratios, risk differences, incidence rate differences, cumulative incidence differences at a selected time point, excess number of subjects based on incidence rate differences or cumulative incidence differences. And the stratification is available for hazard ratios, incidence rate differences, and risk differences. And after calculation, you can either send it directly to the app or save the results as a CSV file to have it available at a later point in time. Okay, so as mentioned, this direct derivation of the CSV file based on CSIS datasets, ADT and TSL, is currently still a beta version and marked like this in the app, with being available just in a different branch on GitHub. So please, if in case you want to use it, please use it with caution. We have dimensioned variables estimates available, but it's planned to add a few further ones to be in line with what's available to show in the forest plot. And after we've done that, we will further think and decide whether we will include weighted multi-criteria decision analysis, including simulation of the weights and study estimates, and or we will consider addition of the DOOR approach, including a potential weighting as well. But this is for the future. So when it comes to using Breezy, you can go to this GitHub page. It is available to everybody. You can download it directly from R in case you have the DevTools package installed and install it, and start the package then with library Breezy, and then just run app and you have it up and running. We have some demo data available currently for the CSV file, but we will also implement soon demo data for the ADTTE. And when it comes to the required file structure, this is also described in the data manual tab of the tab and also on GitHub. And in case you have any questions or you try it out and appear any issues appear, or you have use cases you want to work on with, then please either contact me or Miriam. The emails you can see here. Okay, that's more or less it. Just as the last thing, I want to tell you this Breezy app is just one Shiny app which is developed internally at Bayer among several others, which we are usually using when a study is final. So we have two nice apps which are working on adverse events, Detector and the DEPRO. We have a very nice app, Elaborator, working on lab data. We have Breezy, as I've shown you. We have the very sophisticated subscreen app working on subgroup analyses, and finally for patient profiles, Megaplot app. And there are a few further available, and all these can be found on the Biostatistics Innovation Center homepage, and where you can also find the download links. All of them are either available on CRAN and or GitHub. Okay, with that, I'm done.
Ready?
thank you very much for your attention. And yeah, please let me know in case. And in case you don't have, please put yourself on mute.
Thank you very much, Martin.
Yeah.
Now we'd like to move forward with Danae. Danae, can you take over the screen share?
No problem. So let me do that. And can you please confirm, Brian, if you can see my screen?
Yes, we can. Oh,
Okay.
hold on. It just went to the sun. Your GSK had shown up first, and now it looks like we're back to seeing Martin's screen. Martin, can you
Oh.
stop sharing, please?
You sure?
I see the GSK screen.
Oh, you do? Oh, now I got it again. Thank you.
Okay, thank you for that. So, hi everyone. My name is Danae Williams. I'm a principal statistician working under GSK Vaccine Statistics, and I'm also a member of the BRATS and BRAP task forces as well. And I'm here to present to you a benefit risk assessment example that was piloted on one of my previous oncology studies using the quantitative models CUI and PMCDA. And these models were performed to support the go, no go criteria in choosing the optimal dose for the future oncology studies. And here, this is just to give a disclaimer note for everyone's awareness, of course. And then I'll move on towards the next slide. To give some background, the example that I'll be using is a fictive oncology study, let's call this study B, where this was inspired by a real case where it was a phase 1 randomized single-arm multi-center study. And the study evaluated the safety of the combination of the varying dose of product W, and then you have, of course, the constant doses of products X, Y, and Z, where you have it in adult patients who are transplant ineligible, newly diagnosed multiple myeloma. At that time, we were exploring 5 dose cohorts with the varying dose intensity of product W. And the aim is that out of the 5 dose cohorts, only one of the doses would move forward towards the future planned phase 2 study. So the questions we had at that time were, what is the optimal dose of product W that met that go, no go criteria? And if there were more than one dose that met the criteria, then which dose had the best benefit risk balance? The diagram here below shows you a high-level overview of the steps taken to apply the benefit risk methods in the oncology dose finding studies, in this case A and B. So going from the top, we had the information taken from based on product W's target medicine profile and its value tree. And we move on to the research question, if you look on the left, where the question is, what is the optimal dose for the future studies? And to address that question, we needed 3 key pieces of information, which is 1, the varying dose levels of product W from these single arm oncology studies. And the other piece would be to define the benefits and risks, where the benefit is the proportions of patients that achieve the very good partial response or higher, or VGPR. And then we had the risk, which is the grade 3 or higher product W related adverse events, or AEs, and the grade 3 or higher neutropenia. And the last piece of information was getting the data sources based on the interim analyses from studies A and B. So the analyses were performed using a qualitative and quantitative benefit risk techniques, where we had the CUI model being applied to, along with the metrics that represents the percentage of patients with these attributes. Then we applied the Bayesian probability of success as the estimation techniques. And then we have the weight elicitation that was used as our utility survey techniques. And then finally, we perform a sensitivity analysis based, such as using the Norton and Forest plot for visualization, and the PMCDA model that captures the uncertainty within the data. So here again is just showing you the summary of the methods that my colleague and I had explored within our piloted studies A and B. However, for the purpose of this presentation and what I'm allowed to disclose, what I'll do is I'll be focusing on the CUI and the PMCDA models that was performed for study B. So one of the quantitative methods that I'll introduce is called the Clinical Utility Index, or CUI. And that's an approach where you're combining your benefit and risk outcome into a single metric by using a utility function. So to break this down, if you take a look at the left, the first step is that you want to define your efficacy and safety endpoints. Then your second step would be to model that relative preferences regarding a drug's efficacy or safety endpoint as well. And then finally, you want to assign the weights to these endpoints. And then putting it all together, you'll have what's called like a clinical utility index score. So here are the two approaches that were applied to determine the optimal dose in study B. The first is the go, no go criteria, where it examines that posterior probability of the efficacy, which represents the clinical activity rate, where we're saying that the probability of the rate of the VGPR plus is greater than 72%, is greater than 60%. The other piece is the safety, where we're saying that the probability of a grade 3 or higher product W related AEs is less than 10%, is greater than 60%. Then we have the second approach, which is the CUI, where that's broken down by one benefit component, again, using that same clinical activity rate. And then you have the two risk components, again, using the grade 3 or higher product W related AEs. And in addition, we're including that grade 3 or higher neutropenia, along with their associated weights as well. So the steps were based on an observed interim data, where the first step would be to select the doses which meet the efficacy and safety go, no go. And if we do encounter more than one dose that was selected, then we move on to the next step, where we choose the optimal dose or regimen based on the maximum CUI score. So here again is what I mentioned in the previous slide, again, defining what that go, no go criteria would be, which again, we want to, which is the posterior probability of hitting that target. And at the same time, if we encounter more than one dose that meets the target, what is the maximum CUI score? So the table below shows you the hypothetical observed data that was based from the interim from study B. And here we have our 5 doses. And of course, your second and third column represents your benefit and risk, respectively. And then, of course, we have the question about whether we had met the target for these 5 doses, which unfortunately, in this case, we have not met that. But for the CUI, where the team was interested to see what that score would look like, this was also calculated where it seemed like that the maximum CUI score would be dose 3. But again, because we have not met the go, no go criteria, just due to the fact that we haven't met that go target for the risk in this case. So just to give a quick takeaway message, from my experience when performing with the CUI model, it does take time to define the benefit and risk endpoints to include in your model. And when it comes to the weight elicitation, performing the operating characteristics do help in terms of looking at the different weight options as well. In this case, when you have more than 3, endpoints, it can be interpretation can be tricky, but overall, it is useful when differentiation is critical to success. And again, although it's time and resource consuming, it does allow for like a clear and transparent decision making as well. Lastly, for the sensitivity analyses, this was performed using the probabilistic MCDA, or the multi-criteria decision analysis model, by Waddingham and other authors back in 2016. You'll notice that the framework of the model is similar to the CUI model, but the only difference is that if you take a look at step 2, where you have it, where it's mentioned for the best and worst preferences, this is represented as a random variable rather than back with the CUI model, it was using a single summary values instead. And here, this is just to show you the results of that sensitivity analysis when performing the PMCDA. Again, we're using the same benefit and risk as well with their associated weights, similarly what was used in the CUI model. And if you take a look on the left on this graph, we explored one scenario, which is the extreme case, where we treat the best preferable values as that all the patients had received the benefits of achieving that VGPR higher, but no patient had experienced any of the risk. Whereas if you look at the worst preferable values, it's the opposite, where we're saying there are no patients that achieve a VGPR higher, but all the patients had experienced the risk as well. And how the graph is interpreted is that the higher your PMCDA utility score is, along with its tallest density peak, then that would be deemed the optimal dose you may want to move forward with, which in this case, this would actually be dose 3 that's represented by this purple distribution. And if you take a look on the right, this was another scenario that we explored, in this case, aligning towards the product W's target medicine profile, where we're saying that the best preferable values would be at least 40% of patients had achieved a VGPR higher, and again, no patients experiencing those risks. But the worst preferable values would be the opposite, where again, no patients experience or achieve that VGPR higher, that at least 10% patients or higher had experienced either of these risks. And in this case, what they're deeming to be the optimal dose moving forward would actually be dose 5, which is represented in this blue curve right here as well. So just to conclude as a takeaway message, it does require elicited weights and utilities from other individual stakeholders. And based on that weight elicitation for study B, key stakeholders were very confident in choosing their weights. And when having alongside the target medicine profile and the MBMA models, that also helped to define the utilities as well. It is a multi-step complex process. Interpretation may be less straightforward, but from what was experienced with this model, it seemed that it can be also used as a sensitivity analysis. That way you can access the impact of the uncertainty within your data or model in the case where you have missing or limited information of your attributes. And it can also be used to explore the varied treatment performance as well too. So I think my last slide is just references that was used to support it. And I think that's the end of my presentation. Thank you.
Thank you, Danae. I'm going to share screen now for our panel discussion. I also wanted to remind folks that please feel free to put any sort of questions that you have for discussion in the chat.
Thank you, Brian. I'm Susan Mayo, co-webinar lead for these scientific webinars from our safety working group. I invite our panelists to come off mute and come on camera. Tim Sullivan, Mike Hollaby, and Hong Yang. And thank you very much to Martin and Danae for your great presentations. So we've talked a little bit, those of us who are presenting this information to you today about who will do what and when. I don't think we've talked about which panelist might want to speak first, so I will leave that to Tim, Mike, and Hong to decide who'd like to get started. These are the same questions we had at the May webinar. And so hopefully panelists, you've had some time to think about which of these questions you might like to answer, especially with respect to the presentations we heard today.
Okay, thanks, Susan. I don't mind starting. Are you able to hear me okay?
Yes.
And thanks again for the reference to these questions on the screen. And also to Martin and Danae for the fantastic presentations. I have a question for each, but let me just start with Martin. We know that it can be difficult to implement an organization-wide benefit risk framework. The one that you presented looks quite good to my opinion. Here's the question, just at a high level, can you characterize one of the things that you think has been most helpful that you've achieved with the framework within your company? And similarly, what do you think the biggest challenge has been or continues to be to execute this framework?
So I start with the second one. I think the main issue is still acceptance and avoiding redundancies, because the idea is that we take out of the Cobra the elements and fill up the other documents like CCDS or submission dossier or whatsoever. But in reality, it's sometimes the other way around. So this is, I think, based on the first element, the acceptance. So sometimes this is not there yet in all projects, and therefore people sometimes stick to the old ways, so to say. And please, again, repeat the first question.
It was the opposite. It was, what do you think has been most successful?
Okay. So, I mean, the most important thing was that people start to think about it. So of course, there were these EMA work package reports and FDA benefit risk framework, but this now brought the thinking about benefit risk more into place when it was set up. And I mean, it was mainly driven by PV, and actually by one of the key persons was Leo Pluff, who's now also part of the Science Group. And I think they implemented the BRATS, which is, I think, similar in a way to what we are having. I think this is the main most important thing, that people start to think about it and that there's like a frame to put the thinking together.
Well, that's appreciated. I think what you described are observations that most of us in pharma have experienced doing these frameworks.
Yep.
And in my view, I think it's helpful for our FDA colleagues to hear this. So there's a lot of good intentions in the industry. Sometimes it's hard to get it all pulled together in a large and sometimes bureaucratic organization. And generally speaking, at least in my own view, the more detailed guidance that we can get from the regulators, it helps those of us in industry to pull all this together. Let me squeeze in one more quick question, and then I'll turn over to the other panelists. And Danae, this is for you. Can you give us a practical, real-life example of how you achieve the weighting exercise at your company?
Sure. So this was a bit simple, Tim, because in this case we had a couple of meetings with the stakeholders. In this case, this was represented by clinical and safety, and we had the help of our pharmacologists as well too to help define in order to help also to shape up with the thresholds as well to help generate both for the CUI and the PMCDA model as well. So it was more towards like an elicitation exercise that we had with much further discussions to really be able to understand from the team um, what their expectations are when it comes to the benefit and risk attributes.
Okay, I appreciate that. And when you're doing these elicitation exercises, are there any specific or particular challenges that teams have reported back to you?
Um, I would think maybe the challenge that I mentioned before was really the timing of it, because to really be able to get everyone, sitting together within the call to, work together on the exercise. But I also think the challenge was also mentioning about the weights, which that was a bit of a challenge as well to the team, which initially um, it was a bit of a struggle to really help define those weights, and having to go through various, I guess, operating characteristics or weights to show to the team on whether, does this make sense really? So I think in general, it was really I guess the elicitation was really the challenging part, to really work with the team to defining the weights. And I guess it's the timing as well too.
That's helpful. And then just one more quick follow-up.
Sure.
When you've gotten these weights pulled together, what's been the acceptance internally within your organization? Have you gotten challenges from cross functions and decision makers about the validity of the weighting, or has it been generally accepted?
It's more of the latter, Tim. So it was generally accepted after we had shown the various scenarios with the weights. And I think in the end, both the clinical and safety teams in this case, were happy with the selected weights that we had. And it was aligned with what the pharmacologists had as well too, when they performed their MBMA models as well.
Thanks a lot.
Thank you.
So Hung.
Okay. So I'm Hong Yang. I'm from FDA CBER. So first, I would like to emphasize again FDA's disclaimer. Everything I say, I express in this panel discussion only represent my individual opinion. It does not represent FDA's policy and don't obligate FDA. So in terms of the first, thank you for both presenter, excellent presentation. And then especially I'm very glad to see both presentation. They are the structure approach for benefit risk assessment. That is what you call qualitative. Also quantitative analysis are all align with FDA's benefit risk guidance and sales report. So I would like to emphasize a few. I think they are consistently see the effort from industry. So one new thing that I can see the life cycle approach. So from Bayer's Martin's presentation, you can clearly see how the benefit risk framework are used in the decision making throughout product life cycle. And there is multiple discipline team involved in those benefit risk assessment and the decision. Also patient involvement, we can see from the second presentation, So you have the weight solicitation to incorporate patient preference, and then structure framework. So in FDA's guidance and also sales report both emphasize majority of the benefit risk assessment, the major structure framework. That is what you call qualitative benefit risk assessment. And then, but the quantitative benefit risk assessment can be very variable in some cases if the problem is complicated. But there is no how to say single approach for quantitative analysis. So that is really depend on your challenge equation. Your analysis, quantitative analysis, should be designed to answer that critical question. So there's no unified approach for all the quantitative analysis. It really depends on your question. So more specifically, for example, in some situation, maybe uncertainty is the major thing. Uncertainty of the benefit evidence or risk evidence. In that case, maybe sensitivity analysis or the other approach to address uncertainty is more important. And then in some cases, maybe the preference weight are very, the decision benefit risk decision are sensitive to the preference weight. In that situation, maybe you need to think about to incorporate some weight, like for example, use MCDA. So, but this is not uniform. It really depends on what is your major challenge when you're making the benefit risk decision. So I'm very happy to see all of those are emphasized in both FDA guidance and sales report, also reflect in the industry effort. Let me see what is the the second question, how those can assist FDA review. Like I say early, if there is challenge issue, whatever the sponsor did will be helpful. But for FDA, it does we do emphasize in the guidance, the quantitative analysis is only to support structure framework. We are not using quantitative analysis to replace structure framework. And the quality of quantitative analysis depends on your methodology to answer your challenge question. And also the data quality are very important. So that is something we should consider. Yeah, so I think this is my comment on those presentations. Thank you again for both excellent.
Thank you, Hong. Thank you, Tim. Mike, would you like to offer your
Susan,
comment?
can you bring up my 2 slides?
Oh, Brian, do you have those available?
Oh,
I do.
There we
Here
go.
are.
Excellent.
Thank you, Brian. Yeah, I've been looking so forward to these 2 presentations, and particularly with the PMCDA, we've seen it within our own company this week, a nice presentation, and to see it again, I'm hoping that people in my company will say, okay, this is this is serious. This is what other companies are doing, and you can see the enthusiasm for it. So I have a question here. We've seen a lot of approaches. And so my question is broader. It's to everybody involved in this initiative. And that is, can we create a flow of benefit research questions such that one approach builds on what was learned in the prior approach? Because I see all of these approaches building on each other. Each approach adds its own value to understanding the benefit risk puzzle. And I do see it as a puzzle, bunch of puzzle pieces. Each approach is a puzzle piece. And then how do we go from a forest plot to more sophisticated multi-criteria decision analysis, removing uncertainty each step of the way? So you saw Martin start with the forest plot, and you saw Danae end with the PMCDA. And the question is can we go through steps from one to the last? You would certainly wouldn't want to jump from a forest plot to PMCDA. Next slide, please. And again, these are just my opinions, and everything I said has been published. Okay, so if you're trying to look at these approaches and you're thinking, oh, which one do I want? And you've been doing the forest plot, but you think, wow, that PMCDA captures it all, you know? Well, first of all, the approach that you take is going to depend on the type of data you have. And so I went ahead and just put in here the three types of data. You have treatment level data, which are your summary statistics. Patient level data means you have access to the Adams data, and you're looking at each individual patient, what their benefits and risks were. And then you have the preference data, which leads to the quantitative assessments. And so each types of these data answer questions. And it seems to me that there's an ordering of these questions because each one builds on the previous one. So when I look at the PMCDA that Danier presented, it could really benefit from what Martin mentioned, the door approach. So if you're familiar with the door approach, it's built off of the Norton plot, and it looks at each patient as to whether they had a benefit, no AE, all the way to having an AE and no benefit, and so forth. And you wouldn't decide, do I want to do MCDA or do I want to do door? Well, decision science tells you if you have a choice between A and B, do both. You want to do both because they answer different questions. One thing that MCDA doesn't do is it doesn't look at the correlation between the decision criteria. When it brings in the preference weights, you might not have addressed whether you have what they call preference independence, which is what the experts who do preference studies, look at. So all of these things building on each other. And I love the presentations that have been given in this two-part series, but I'd like to roll all this back to everybody involved in this initiative, both for the BRAT and the BRAP. Can we create a flow of research questions that should be answered so that when you get all the way down to that PMCDA, that you've done all your homework? Because you can actually get counterintuitive results from the more sophisticated quantitative approaches if you haven't answered the questions up above, like particularly with the patient level data. You haven't answered, you might have a drug that falls in the on acceptance region. You see in that first trade-off plot where the acceptance region, you could have a drug that doesn't fall in the acceptance region. If that's the case, you might want to know that before you go to more sophisticated approaches. So each of these, for example, MCDA doesn't tell you whether the benefit risk profile is sustained across time. If you look at that third graph there, it shows you there that the benefit actually increases over time and the risk actually plateaus. That's like perfection. You want that. These are the types of questions you have to be answering along the way before you get to the more sophisticated approaches, so that it builds more trust, and it also shows you the added value of each approach as you go along the way. This circles back around to Tim. Tim is asking, what is the uptake of these approaches at your company? The uptake at your company is going to depend a lot on you demonstrating the added value of each one of these approaches. And as statisticians, if we can't show the added value, we can't really fault our organizations for not giving them much attention. So that's my 2 cents. And I kind of like to hear from others.
Thank you, Mike. I just wanted to tie one of the comments you made in with Hong's, where Hong was talking about that the quality of the data and the analysis will have an impact on the assessment. And what came to mind as I listened to both of your comments is, does that also depend to some extent on what the clinical question is that you're asking in terms of, is this drug a new drug, a drug for an unmet need where there's no other drugs available for that indication, or is it maybe a third or fourth drug in a group of drugs that are already approved for that indication? What the safety profile is versus maybe other options for patients, as well as the efficacy profile. Do either of you have thoughts on that?
Well, people think that we're only trying to answer one question: do the benefits exceed the risks? But that's the granddaddy of benefit risk questions. There's a lot of sub questions that lead up to it. And so when I watch a lot of presentations on methods, I always like to see the question that it's answering right there at the top.
Agreed. I have a lot of influence for me on use of the estimand, where the clinical question is also of ultimate importance. And maybe that's true for any kind of structured framework, that we need to make sure we keep the question in mind. Hong or other panelists or speakers, do you have comments on this or other comments panelists have made?
Yeah, I can make one additional comment. Is I went to several of the conference workshop present. So there is a lot of discussion. There is many questions discussed about quantitative approach. So some people say, oh, shall we propose we do MCDA for all the benefit risk assessment? So that is just my personal opinion based on my understanding with participate of SIOM's working group and FDA's guidance working group. I don't think MCDA should be applied for all the benefit risk assessment. So one thing is really depend on your question. Some benefit risk assessment is not is not the major question. The major question is how, for example, like in FDA, we did a benefit risk assessment for COVID-19 vaccine. We don't use MCDA. Why? Because at that situation, it's not the outcome. The way of the outcome is not the major challenge. Major challenge is how do you translate clinical efficacy to the real world evidence of the benefit and compare to real world risk. So in our benefit risk assessment, we don't use MCDA. Instead, we use a modeling approach, try to estimate how is the real world benefit in terms of how many cases are prevented. So because how many cases prevented is not just depend on vaccine efficacy you got from clinical trial. It also depends on many factors. For example, disease incidence among the population. When the pandemic changed, the disease incidence is changed. There is a lot of uncertainty. And then also the new virus keep emerging. And the vaccine efficacy present in the clinical trial may change in the real world. So those are also uncertainty. So our modeling benefit risk assessment mainly focus on to address those uncertainty. The way is not that important. When we address those uncertainty, we put out the results, say how many cases will be prevented in terms of how many risk will occur for the myocarditis cases related to the vaccine. When we produce that result, and then the reviewer very clear, benefit is always the risk. They don't need a wait COVID-19 cases and the myocarditis cases related to vaccine. They don't need that. So MCDA in that situation is not necessarily a right approach. So that's my opinion is quantitative analysis, there's no uniform approach. It really depends on your major challenge equation. So you need to use the approach to address your challenge equation.
It's helpful to hear FDA's perspective on that. In my view, what you're describing with the vaccine example is still a quantitative approach. It's just a simpler one. It's basically a numbers needed approach or some extrapolation of that. What would help, first of all, doing MCDA, even for large companies, can be daunting, and there's some resistance to doing it. It would help if we could all agree when it's appropriate. To a first approximation, I think it's appropriate when you have a borderline overall benefit risk and multiple competing key benefits and key risks. That's my simple version of when I think it's appropriate, and I would welcome the view of others. And then to finish off, to go back to Mike's comment, I think it's a fantastic observation, Mike. What I'm hearing you say in the simplest terms is a garbage in, garbage out phenomenon. You know, you can plan some fancy quantitative analysis, but if you haven't taken the steps to think through your methodology and assumptions first, it's not very useful. So I agree with you. If there could be some kind of position paper or thought on the steps that you outline, the predicate steps before you get to your MCDA, that would seem to bring a lot of value. Maybe for an interested and capable person, it could be a manuscript.
Yeah, well, I think we're talking to the people that could make that happen. Statisticians know that there's no free lunch. Every method comes with assumptions and uncertainties that have to be addressed. And I think that's what we do best. And so as you go down this list, you're doing your homework. You know, you're addressing assumptions, you're chipping away at the uncertainties, And I think then you can see there might be a stopping place for your particular project where you say, we've gone really as far as we need to go. I think we have all the puzzle pieces we need, but we can still do more. But if you do a leapfrog and you go from the top to the bottom and you get a counterintuitive result, I'm afraid it's going to shake the confidence with our colleagues because they'll sit there and go, well, that doesn't make sense. And you sit there and go, oh, wait a second, I need to back up and do my homework. I need to go back and maybe look at that trade-off plot and understand, the trade-offs with something, particularly that trade-off curve where you're seeing, patients are willing to accept more risk if they can get more benefit. But another method ignores that. And so,
Yeah.
Yeah.
Mike, I would have to agree with Tim. When I saw your slides in my inbox this morning, I too was thinking, oh, maybe Mike will write a manuscript or ask others to join. Because I think the more we characterize these things, the clearer it becomes to people how the different pieces fit together for those people who have more experience with it. I see a couple of questions in the chat. Are there any more burning comments before I go to those questions? Okay, so Mustafa Nasser says, thank you so much for the presentations. I was just wondering, how will you approach, this is for Martin, how will you approach including patient-related outcomes in the Breezy app in the final assessment?
Yeah, so currently the only way we can approach it is, since we are doing semi-quantitative analysis, to sort the outcomes by the assigned weights. What we plan in the future is to include an MCDA approach, including simulation of weight data based on a distribution, and similarly of the outcome study data. We've done this previously in other projects, and I think there's also a publication available, or at least a presentation from a DIA workshop, I think, from last year. But yeah, this is what we plan but have not started yet. I mean, there's R code available to do this internally, but it's not implemented in the app yet.
Okay, good news to know that's in the pipeline for you, Martin.
Yeah.
And then Susan Kalila asks Danae, who were the stakeholders who provided the preference weights in the quantitative analyses? And what type of methods and surveys were used to ascertain the preference data?
Thank you, Susan. And to answer the other Susan's question as well. So the stakeholders that were involved with the preference weights, that was the clinical and safety leads for that study. And we did actually had some help with the pharmacologist in terms of helping to define the thresholds. But and we did perform again the operating characteristics through simulation to see whether if they, do you want to see this happening more like at a 60% or 70% and so forth. So hopefully that addressed the first question. And your second question about the methods that were used to ascertain the preference data. So we didn't have a specific method or, a statistical technique that was used to capture the preference data. This was more towards like we just used an elicitation in this case with the stakeholders to really ascertain that preference data you mentioned. But hopefully, Susan, that answers your question.
And Susan Kalila, if you're still here, if you had
Yeah.
any
Hi,
follow-up.
yeah, thanks. I was just wondering if did you do it via like a survey of some sort, or you know, sort of a trade-off, or with quantitative numbers? I was just a
Oh,
little
sure.
curious how you did that.
Yeah, it's more towards, I guess, the quantitative numbers that we shared with the stakeholders, you know, going back and forth in terms of really asking them the questions in terms of, you know, what would be defined for their benefit and risk. And then for the numbers that they see, like maybe in this case, they want to see a range between this lower limit, this lower number and this higher number. We generated some simulations and showed them what the results may look like before we had the actual data.
Okay. Okay. Thank you.
Thank you.
Can I
Oh,
ask
yes,
a question related?
sure.
So you mentioned about when you determine the way you consider operating characteristic. Can you explain a little bit what kind of the operating yeah.
Sure. So, Hung, what we did was using the in the beginning, what we did was we used different weights as the operating characteristics. So in this case, we had the threshold thanks to the pharmacologist when they performed the MBMA model. The question was to the team as to how much weight you want to apply to for their benefit and also with the risk as well. So that's where the operating characteristic piece came in, where we had various weights for the benefits, various weights for the risk, and combining that together to see what kind of, you know, their expectation to see what they would like to see with the CUI score, for example. But that's what I mean in terms of like the operating characteristics, where we were looking at different weights applied to these attributes.
Oh, okay. Thank you.
Thank you.
Okay. Brian Waterhouse, did I miss any questions in the chat?
No,
I
you
have
didn't.
a
Oh, but there's another
ha-
question.
All
Yeah,
right, Hans.
I have a question for Martin. So I think Martin presentation mentioned like when you say when you conduct semi-quantitative, when you have a similar type of benefit risk outcome, can you explain a little bit what you mean by the similar type of benefit risk outcome?
Yeah, it's similar in severity. I mean, the standard example is the anticoagulants we were working on. I mean, there you have hard outcomes for efficacy and safety, meaning strokes, MIs, and so on the one hand, and bleedings on the other side, while we had other compounds where we have efficacy measured by 6-minute walking distance, and safety concerns were from minor to severe issues. So yeah, there it's more difficult to do a semi-quantitative analysis, and there we rather look into a qualitative assessment.
Thank you.
Since we have a little time, I'd like to ask Martin a question too. In a previous life, Martin, I worked on creating benefit risk forest plots as well, not as sophisticated as yours, but we considered using different axes for the different endpoints. In other words, not turning everything into one measure, but leaving them in the units that the clinicians were already familiar with. And while that maybe makes for a more complex looking graph, it does leave those units in a form that the clinical folks who are generally used to seeing things in that format can read. And we realize that clinicians all of the time have to look at different units and in their mind have to weigh what the benefits and risks are. Were there any discussions along that line as you were creating the Breezy app with your colleagues?
Internally, not. I mean, we had this discussion when starting the benefit risk assessment tool suite, because I mean, there are pros and cons for both approaches. I think you very nicely described the pros for your approach. I mean, maybe because I'm educated in this anticoagulant setting. For me, it was always important when I do a benefit risk assessment analysis that everything is done on the same scale. So for me, this is the additional or one of the additional things, also removing double counting when you do the benefit risk analysis with the so to say added value doing this analysis compared to analysis you do on the CSR level, doing analysis for efficacy and safety separately. So therefore, this was always my thinking, to bring everything on one scale, use the same, in the old days we called it ITT approach or treatment emergent approach. Now you would say you use the same estimate for efficacy and safety. But as said, there are pros and cons to both approaches, I think.
Mm-hmm.
That's right. Well, and Susan, you know, that you actually highlighted one of the difference between directions. Probably not surprisingly, since I grew up in the same place as what you're talking about. I was also thinking about for the BRATS tool, that the benefit risk forest plot would allow for different graphs for different statistical measures, right? So, so that you can keep those units live. But this is a place where the forest plot approach for BRATS may be different from Breezy, and why there might be a reason to have both tool suites available to people. But I think that's an interesting further discussion that we should have within the task force on whether or not, you know, what is the general thinking about the pros and cons of each and which direction we want to go as a task force.
Oh, that's good to know, Brian. And yeah, I don't have again, this is just my personal opinion, but I think if as a group there's, you know, the general direction is seen as converting everything to one single measure, I think that's reasonable as long as that makes sense to the people who are needing to make the decisions based on that information.
Yeah, but I think we should follow up at the task force level for that one. Yeah, but I agree with your opinion.
Okay,
Who's the end user, right?
that's
and what
right.
makes sense to them. Yep. All right, we only have about a minute left, so I think I would like to end here with thanking very much Martin and Danae for your wonderful presentations. Thank you to Tim, to Mike, and to Hong for the great panel discussions. Thanks for Susan and Michelle and Meng Chun for helping to organize this. And of course, Susan, especially you helping with the panel discussion and running this meeting today. And last, but certainly not least, thanks to everyone here in the audience with your participation, your discussion points, your questions, and your attentiveness. So thank you very much. This ends the meeting today. Have a good rest of your week.